Chapter 5.2 Carbohydrates serve as fuel and building material. Carbohydrates include sugars and their polymers. The simplest carbohydrates are called monosaccharides, or simple sugars. Disaccharides, or double sugars, consist of two monosaccharides joined together by a covalent bond. Polysaccharides are polymers of many monosaccharides. Sugars, the smallest of the carbohydrates, serve as fuel and a source of carbon. Monosaccharides generally have molecular formulas that are some multiple of the unit CH2O. For example, glucose has the formula C6H12O6. The monosaccharides have a carbonyl group and multiple hydroxyl groups. Depending on the location of the carbonyl group, the sugar is an aldose, or an aldehyde sugar, or a ketose, a ketone sugar. Most of the sugars, their names end in the OSE ending. That's how we can identify them very quickly. Monosaccharides are also classified by the size of the carbon skeleton. The carbon skeleton of a sugar ranges from 3 to 7 carbons long. Glucose and other 6-carbon sugars are called hexoses. 5-carbon sugars are called pentoses, and 3-carbon sugars are trioses. Here we can see the aldose and ketose version of some trioses, some glyceraldehyde, dihydroxyacetone. Here we have pentoses, we have ribose and ribulose. Here we have hexoses, 6-carbon sugars. Notice the similarities between glucose and galactose. The ketone sugar is a fructose, which is fruit sugar. Another source of diversity for simple sugars is the spatial arrangement of their parts around asymmetrical carbon atoms. For example, the glucose and galactose, both 6-carbon aldoses, differ only in the spatial arrangement of their parts around asymmetrical carbons. Although glucose is often drawn with a linear carbon skeleton, most sugars, including glucose, form rings in aqueous solutions. Monosaccharides, particularly glucose, are major nutrients for cellular work. Cells extract energy from glucose molecules in the process of cellular respiration. Simple sugars also function as the raw material for the synthesis of other monomers, such as amino acids and fatty acids. Here we can see the linear and ring forms of glucose. Notice the numbered carbons. 2 monosaccharides can join with a glycosidic linkage to form a disaccharide via dehydration. Maltose, or malt sugar, is formed by joining 2 glucose molecules. Sucrose, or table sugar, is formed by joining glucose and fructose. Sucrose is the major transport form of sugars in plants. Lactose, or milk sugar, is formed by joining glucose and galactose. Think of it as when we take a water molecule away from the sugar, it makes it stickier and they stick together. And that's how we form those disaccharides. Here we can see examples of disaccharide synthesis. Polysaccharides, which are the polymers of sugars, have storage and structural roles. Polysaccharides are polymers of hundreds of thousands of monosaccharides joined by glycosidic linkages. Some polysaccharides serve for storage and are hydrolyzed as sugars are needed. Other polysaccharides serve as building materials for the cell or the whole organism. The architecture and function of a polysaccharide are determined by its sugar monomers and by the positions of its glycosidic linkages. Starch is a storage polysaccharide composed entirely of glucose monomers. Plants store surplus glucose as starch granules within plastids, including chloroplasts, and they withdraw it as needed for energy or for carbon. Animals that feed on plants, especially parts that are rich in starch, have digestive enzymes that can hydrolyze starch to glucose, making the glucose available as a nutrient for cells. Grains and potato tubers are the main sources of starch in the human diet. Most of the glucose monomers in starch are joined by 1 to 4 linkages, which just means that the number 1 carbon is joined to the number 4 carbon. The simplest form of starch, amylose, is unbranched. Branched forms such as amylopectin are more complex. Animals store glucose in a polysaccharide called glycogen. Glycogen is similar to amylopectin but more highly branched. Humans and other vertebrates store a day's supply of glycogen in the liver and muscles, hydrolyzing it to release glucose to meet the body's demands for sugar. Cellulose is a major component of the tough walls of plant cells. Plants produce almost 100 billion tons of cellulose per year. It's the most abundant organic compound on Earth. Like starch, cellulose is a polymer of glucose. However, the glycosidic linkages in these two polymers differ. The linkages are different because glucose has 2 slightly different ring structures. These 2 ring forms differ in whether the hydroxyl group attached to the number 1 carbon is fixed above or below the plane of the ring. The differing glycosidic linkages in starch and cellulose give the 2 molecules distinct 3-dimensional shapes. While polymers built with alpha glucose form helical structures, polymers built with beta glucose form straight structures. The straight structures built with beta glucose allow hydrogen atoms on one strand to form hydrogen bonds with hydroxide groups on other strands. In plant cell walls, parallel cellulose are held together in this way, and they're grouped into units called microfibrils, which form strong building materials for plants. Cellulose microfibrils are important constituents of wood, paper, and cotton. Here you can see the structural linkages of the alpha and the beta glucose monomers. Starch is a polysaccharide of alpha glucose monomers. Cellulose is a polysaccharide of the beta glucose monomers, making every other glucose monomer upside down with respect to its neighbors. Enzymes that digest starch by hydrolyzing its alpha linkages cannot hydrolyze the beta linkages in cellulose. Cellulose in human food passes through the digestive tract and is eliminated in feces as insoluble fiber. As it travels through the digestive tract, cellulose abrades the intestinal walls and stimulates the secretion of mucus, which aids in the passage of food. Some microbes can digest cellulose to its glucose monomers through the use of cellulase enzymes. Many eukaryotic herbivores, from cows to termites, have a symbiotic relationship with cellulose-digesting prokaryotes and protists, providing the mammals provide the microbes a safe place to survive, and then the host animal receives access to a rich source of energy in turn. Some fungi can also digest cellulose. Another important structural polysaccharide is chitin. Found in the exoskeletons of arthropods like insects, spiders, and crustaceans, chitin is similar to cellulose, only it has a nitrogen-containing appendage on each glucose monomer. Pure chitin is leathery, but can be hardened by the addition of calcium carbonate. Chitin also provides structural support for the cell walls of many fungi.